Hello, it's Eric Topol with Ground Truths. And with me today is Professor Shane Crotty from the La Jolla Institute of Immunology, not too far away from where I work at Scripps. And Shane has been a go-to immunologist colleague here in the Mesa. And he and his colleagues were the ones that really first published the response to SARS-CoV-2 as far as the immunologic response. And today we're doing something very unique. We're going to go over, for the first time in the 2-year plus history of Ground Truths, going to have a publication with at least simultaneous or near simultaneous podcasts. Shane, so welcome and congratulations on this really important paper in Nature.
Thanks, Eric. Thanks for having me. Yeah, somebody asked if I was going to go over to Scripps for the podcast, and I was like, "Yeah, we could."
You could, but no, it's good and it's nice having the logo of this great institute you work at right in the right corner, and you've done so many contributions with your colleagues at the Hoy Institute. It's really a privilege to have a chance to learn from you, and particularly about what we're going to talk about today, which is mucosal immunity to upper airway infections, which is especially germane to COVID. And we're actually in the middle of a significant wave of COVID right now. And I guess it would maybe be fair to say, Shane, that we've never truly understood the underpinnings, the real details of upper airway mucosal immunity. Is that a fair statement?
Yeah, it is a fair statement.
Okay, so today we're going to crack the case. This paper from you and your colleagues, of course, you're the senior author and first author, Sidney Ramirez, did a remarkable study. I mean, just extraordinary. This is why we're doing a special podcast about it. Maybe you could just kind of give us the overview of the design, because you were doing things that haven't been done before.
Sure, and I'd say the genesis even of it goes back to what you were introducing. I mean, during the pandemic, we, like a lot of scientists, spent a lot of time and energy trying to help understanding immune responses to this virus and immune memory to this virus and what was involved in protective immunity. And we're certainly proud of the work that we did, and it was hard work. And after a while, we were exhausted and we stopped. um, and then we came back to it after a while, and said, well, the virus is still here. And so many people have contributed so much to better understanding the virus and creating vaccines. But there are clearly still things we don't understand. What are those biggest knowledge gaps, and where might we be able to contribute? And really, to me, the biggest one was location. That this is a virus that and infects your nose, infects your upper airways, your nose and throat and oral cavity, and then obviously if you get severe disease, the severe disease and death are from the lungs. And it's just been a big knowledge gap in terms of understanding what actually occurs in those tissues immunologically, and what is associated with protective immunity, or what could be associated with protective immunity, and sort of looking forward, um, what might be helpful for mucosal vaccine development, from things that we could learn. So we started from what we would call the basics, right? And what does immune memory look like in the upper airways in normal people? And that hasn't been available, really, even in, when we started this 2 years ago, even in the biggest atlases published of the human body, right? There was no upper airway tissue representation at all. And that's because technically it's just tough to access and difficult to reproducibly get at. And so we recruited people, to a group of 20 to 30 people, to come to LJI once a month, and just started testing out published and unpublished sampling techniques to see were there ways where we could reproducibly sample immune cells in the upper airways from people. And once we um, got things. So the keys for us were you got to have enough cells that you can collect, to learn something from. And luckily, with modern techniques of flow cytometry and single cell sequencing, you don't need that many cells. And so we could get 100,000 cells on a swab, and that's enough to do a lot with. And second, how reproducible was it? So we showed we had people come in every month for a year, and we could reproducibly find the same things in their swab, same cell types in their swabs. And the third thing was that people would come back. um, we found that if you have uh, good nurses doing the techniques, we could find ways that this would be a sampling approach that was tolerable, and people would come back for repeat measures, which is really valuable, right? To see what's happening in people over time. So that was what we started from in the study and built from.
And if I correct, you sampled two places with the swabs, one in the nose and one in the throat, or I think one which was what you have in the paper as the MT for something about the median nasal turbinate, and the other adenoid in the back of the throat. Is that right?
So all the sampling is a swab into your nose,
Okay.
and when we were doing that, um, we were really excited to see the diversity of immune cells, particularly T cells and B cell memory T cells and B cells that we were isolating. And they're like, wow, there's actually a lot of interesting immune memory up in there. And the lab said, oh, by the way, we're seeing T follicular helper cells. Now that happens to be my favorite cell type. Uh,
Why is that, Shane? Of all the cells, why do you say that's your favorite?
so,
I
so
know you published a lot on it, but
So those are the T cells that are required for basically all neutralizing antibody responses. All high quality antibody responses depend on almost all high quality antibody responses depend on T cell help. That T cell help comes from T follicular helper cells. So
okay.
all antibody evolution is certainly one of the coolest processes of the immune system, and all of that depends on T follicular helper cells. So the fact that, for example, you could get Omicron neutralizing antibodies even after only being vaccinated with ancestral vaccine, right? That's the immune system making guesses
Mm,
what variants would look like. And those guesses come about through this antibody evolution that's driven by T follicular helper cells. So it's
okay.
really one of the most brilliant things the immune system does. And that's a cell type that's key for it. But those processes happen in lymphoid tissue. That's what happens in lymph nodes and spleen. And here we were sampling epithelium, right? Your nasal epithelium. So the cells didn't really belong there. And so that's what turned the study in another direction. And we said, okay, let's figure out why is it that these cells are present in these swabs. And we had a couple of possibilities. And one possibility was that the swab was going all the way back to the posterior wall of your nasopharynx, your top of your throat, and sampling adenoid tissue. So adenoid tonsils. And adenoids are a true lymphoid tissue, and they're a mucosal lymphoid tissue. And so we came up with multiple ways to validate that that's what we were testing. And in fact, it was the Sydney Ramirez, a clinician, and the ENTs involved who said, well, let's just look. And so they actually did endoscopies with the swab to actually
Oh
see
wow.
where the swab went. And
Wow.
we've got videos of the swabs going into the adenoid crypt in the back. And then we've got measurements of here are the cells that were, that you find on those swabs. And what's cool about it is that, yes, so we did studies with we then shifted to doing studies with two sets of swabs. One where we essentially went, quote unquote, halfway back, where we're detecting the epithelium of your nasal passages, and then one where it was all the way back and detecting the adenoids, the lymphoid tissue. So here we've got two different sites, right, in your upper airways that are about an inch apart, and we're detecting essentially completely different cells of the immune system at those two places. And we tend to think of the cells present in that epithelial tissue as probably the sentinels, the cells that are sitting there that can potentially immediately reactivate, right, and try and protect you against a viral or bacterial infection. Whereas the lymphoid tissue, the adenoids, is really about generating the immune responses in the first place and priming immune responses. And that's where these germinal centers can occur, which are where the TFH are, where you can get antibody evolution. And so we found in the course of the study that with this non-invasive technique that we
Yeah,
can.
by the way, I don't want to be signing up for the one way up there because I mean, just the mid nose is enough for me. So, wow, I got to give credit to your study participants for coming back every month for a year to have that.
12 months.
Some people call it a brain biopsy, but
Right. So I will tell you, it is a different experience than the COVID nasopharyngeal swab you might have gotten through your car window.
okay, okay.
It if you're actually sitting down in a comfortable space and there's a nurse, doing it with these particular goals. We really found we had 100 people in the study and a total of 300 swabs, and the vast majority of people came back if we asked them to.
That's great.
So it is, and we're certainly very thankful for the volunteers. Obviously, they were volunteering in the first place, so to participate. But yeah, it's quite doable. So I'm a little hesitant about the video because I've told people to not show it to potential volunteers because it definitely doesn't encourage you to volunteer. You're like, wait, that's what's happening, but it's actually I've had it done on me. It's all
Not that bad.
it's
Yeah.
it's really pretty compelling. And by doing these repeated samples, so we actually now have the capacity to look at ongoing immune responses, like after an infection or vaccination in people, and see how that results in the immune system changing and what might be the source of the protective immunity that comes up. So we've actually got data in the paper looking at this antibody evolution in real time. So we've got affinity maturation of B cells occurring in just normal, healthy adults of mucosal B cells against COVID, and so that's really helping us learn what's possible, basically, to figure out. Okay, if you're gonna try and make a vaccine, what types of immune cells are even possible to generate in this tissue, and where might you try and generate them? Or if you're trying to study some disease state, what are types of cells that might be problematic?
Yeah, I mean, I think the idea that so many of us have been pushing for a nasal vaccine to induce mucosal immunity, because, as you know very well, the current shots are not very good at any durable or substantial protection from upper airway infections of COVID and or SARS-CoV-2 and other infections. So I think one of the most important parts of this report is that it lends itself well to helping towards artificially, if you will, with a make a vaccine to go with the protective to get the protective features that you were able to identify. Which maybe you could just, if you had the ideal nasal airway, what would the cellular profile look like?
I see. Uh, yeah, great question. Yeah. So first of all, antibodies are great, right? So I've most of my career has been dedicated to most licensed vaccines. The correlative protection is antibodies. Antibodies clearly can be protective, and if you can get them, that's excellent. So certainly, I would want, in terms of the non-cellular component, I would want antibodies present, neutralizing antibodies present uh, in that.
And are these IgA or IgG?
So yeah, in an ideal situation, what would I want? I'd want a mix of both, basically, because
Okay.
the
Okay.
IgAs look like they have a little more protective efficacy, but the IgGs, sorry, just at a molecular level have a longer half-life and
Okay.
stick around a little
Yeah.
longer. So yeah, I'd want both. Um, and then really the premise for most of what we do is saying, okay, in situations where antibody isn't enough, or the antibodies don't stay around long enough, or you've got a variant that now obviates the protective efficacy of that particular antibody, are there other types of protective immunity you can have? And the immune system has other stuff besides antibodies for a reason, right? Like, of the lymphocytes in your blood, most of them aren't antibody producing cells.
Right.
They're,
Right.
most of them are other things. And so, well, sticking with adjacent to antibodies, I would want, um, and so those antibodies in the mucosa, yeah, I'd want them to be made by cells that were literally right there. So plasma cells living in that site, so that you've got basically the highest concentration of antibodies you can get, because they're not having to diffuse through the whole body. They're just already at their highest concentration right there. Now, antibodies come from B cells. That's what encodes the antibodies. And so the B cells can make neutralizing antibodies if it turns out that you haven't made enough neutralizing antibodies, or if there's a variant that escapes those. Uh, Maybe there are other B cells that could make, once you get infected, more B cells that could make more antibody rapidly,
Then,
infection, or B cells that recognize this variant that is mismatched to the current antibodies you have. But memory B cells are basically a library of um, different antibody specificities representing sort of different guesses about what um, viral variants or structures might look like. And so I would want memory B cells in that upper airway tissue that could reactivate quickly. So there are memory B cells in your blood, and we don't know how long it takes. And that's one of the reasons we're hoping we and others build upon this study. But it might take, let's say, 5 days for memory B cells to go from your blood into your upper airway.
oh, right. That's,
And that's right, you're already quite sick by that point. Instead
yeah.
of memory B cells right there, and as soon as virus showed up, right, they got activated. Now maybe after, we're not sure yet, right? But now maybe after 48 hours, those cells are now activated and doing something useful. That would be optimal. So then we can pivot to the T cell side.
Yeah.
Um, So there's a fantastic recognition that T cells being physically present in tissues, tissue resident memory cells, as they're most often called now, can really have fantastic protective capacities from a lot of mouse model systems, where you can see if T cells are in the skin or the liver or whatever already. They're more protective than if the cells are in the blood. So if you could also have T cells essentially permanently parked in the epithelium of your nasal passages and in the um, adenoid, hopefully those could essentially be sentinels for protective immunity. And as soon as you get infected, those T cells would reactivate and start killing off infected cells. And that's the mix that I would want to see. And I think there's at least some reasonable evidence in the context of COVID that people who have um, T cells in their upper airways maybe manage to control the virus so quickly that it's a subclinical infection. They never notice they get infected. And so building on those types of observations, Yeah, that's what I would.
That sounds good. I like that. I'd like to have that in my nasal airway. Now, just to make sure I've got this, which is what you found, of course, the memory B cells, the T cell memory, CD8 positive, that is the cell killing T cells that you mentioned, resident T cells. One clarification on that, they they are not really going to do much until there's been some cells that have been infected with the virus, right? Then they come alive and kill those cells. So they're not an immediate, but they can work pretty quickly still, though, right? If they're resident T cells.
Yeah, in theory, it might take as little as 12 hours for a virus to infect a cell, and then you get some antigen presentation on that cell that could, activate the T cells. It could.
And that's all happening perhaps within the incubation phase of the virus, right? That's
Correct.
right, right. So the other thing,
That's a tough thing to study, but conceptually, that's the way people tend to sketch it out.
right now, the other part of the story is, and you alluded to it earlier, is the lymphoid tissue up there, higher up, where they are these germinal centers. And is there anything different you want in these germinal centers? Do they contribute to mucosal immunity that you haven't already mentioned?
So that they really contribute in this forward-looking sense, or really in the classroom kind of sense. The germinal centers are where you want to be, or where you're basically teaching the B cells in advance of seeing the infection,
Uh,
either with your vaccine or with your previous infection, evolving better B cells and better antibodies, and hopefully instructing them where to go reside to then be ready um, for the next infection. If you get really great protection that next time, hopefully then you don't need to start
right. So
with
it's like the training grounds for this coordinated response, I guess, right? Now, you also noted this. I mean, this is a rich paper, which is, I
that.
mean, we're illuminating something that's never been done before in human beings. I mean, it's pretty damn important and impressive. But you also found that you had an age relationship. Can you tell us about that?
Sure. Uh, this is one of our favorite parts of the study. Uh, I'd say in particularly for several of the clinicians who were involved, because um, uh, the general conversations people have about upper airways lymphoid tissue, like your tonsils and including your adenoids, is that adults don't really have functional lymphoid tissue in the upper airways. That your tonsils atrophy by the time maybe you're 20 or something, and so um, immunologically, functionally, what that means is you go, well, okay, if you have, let's say, an intranasal vaccine, right, or or you get infected with SARS-CoV-2, if those would normally be the sites that start your immune response, where does it now happen? And instead, what we saw was we had um, such a diverse group of people in our studies. We realized we had people from age 18 to 68, and so we could directly ask in normal, healthy individuals across uh, a large age span of adulthood, is there functional mucosal lymphoid tissue? And the answer was yes, it was there, but it definitely declines over
Mm,
time.
mm.
And it's declining, on a log scale. Our simplest statement was that 75% of everybody we sampled still had functional tissue, but the younger the people were, the more functional it was, and the more germinal centers actually we saw, which were again these training grounds.
Yeah, so this is really important because we know for COVID and obviously for influenza and other respiratory infections that people of advanced age are much more susceptible. And here you are finding something that supports that. And it's almost like it's like the thymus, the involutes after that, what age 20, and our lymphoid tissue, we're just, set up to fail. I was just, you know, old codgers like me, we're defenseless, I guess, right?
So, and what I've liked about that in a positive sense is that it's not that all of these things go to zero. Like, for example, naive T cells are definitely less abundant in people over the age of 60 than under, but they're not zero. And the mucosal lymphoid tissue is definitely less abundant in people over the age of 60, but in most people, it still wasn't zero. And I always think about these things from a vaccine immunology perspective, and fundamentally, the difference between getting vaccinated and infected frequently is that the whole point of the vaccine is you get to generate the immune response on your own time, right? And so even if you're starting with 5 times fewer T cells or 5 times fewer germinal centers, if you're getting to do all that training ground in advance, you can end up with a just as many virus-specific T cells as a 20-year-old, or just as many memory B cells as a 20-year-old, because these things occur on an exponential scale, right? Because of the cell divisions, and so you can it might take you 3 extra days, for example, to get to the same level, right? Which again, if you're racing a virus, can be the difference between life and death. But
Oh, yeah.
if it's not a race, right, and if you're doing it in the context of a vaccine, it's a much smaller factor, and that's some of what we've been trying to learn.
Yeah, now we only have started to scratch the surface of your findings. One of the things that drives me nutty in reading papers, especially from great immunologists like you, is that in each figure there's like 20 different panels. We get to panel one of the figures. Figure 3 is all the way to panel W. So I mean, that starts with A. That gives you a little impression of the data. It's rich. Another one goes to N or R. I mean, we're talking about a lot of data. So I've only started to, really deconvolute what you've done here, which is just an amazing study. But what are some other things that we should touch on before wrapping up?
A lot of the goal in this study was to establish baselines of what is normal in humans in the upper airways, and that's one reason why in this case there actually are a lot of figure panels, because we could break things out into a bunch of individual parts of the immune system that really hadn't been characterized in this way before. Um, Something we really cared about was durability of immune memory. It often talked about, well, mucosal responses are inherently short-lived, and we're like, well, what does that mean? Does that mean there's just no memory? Is it different kinds of memory? And so this is the first measurement of memory B cells in this tissue in an antigen specific way. And we were doing it in people who had had recent COVID breakthrough infections. And we saw really the mucosal memory was stable for 6 months. And so to me, that's quite encouraging that, it's not 1 month and it's gone, at least with an infection. It's at least 6 months, and it looks like it'll project out for substantially longer. And amongst those cells, many of them are IgA. So IgA is this antibody isotype that's particularly mucosal associated, and only 5% of the memory B cells circulating in blood were IgA. Okay. Whereas many of the memory B cells in the local tissue were IgA, which we think is also telling us that there's a lot of immune memory and the immune system in this tissue that we're probably not sampling in the blood. And so sampling blood's great, right? It's accessible and we can learn a lot from it, but it does look like there is some tissue compartmentalization.
Oh, without a question. And like the findings you had of the resident T cell is so indicative of that.
Yeah.
And what's really striking, of course, Shane, is that as we assess the immune system, in people at large, we we look at the lymphocyte neutrophil ratio. We get almost nothing. And then, in the course of the pandemic, you and your colleagues there provided such granular data on B and T cells, CD4, CD8 T cells, and the you illuminated things that are not done ever clinically. These are research, high tier research labs like yours. The only question I have on before I just wrap up with the nasal vaccine story,
Mhm.
what interferon wasn't really part of this. And
Correct.
as we know that SARS-CoV-2 can shut down the interferon response, it's considered a frontline part of the defense. Where does that fit into the mucosal immunity of the upper airway?
Yeah, it's really important, and uh, that's in this basic divide we do in the immune system, the innate immune system and the adaptive immune system. So everything I was talking about is the B cells, the T cells, and antibodies. That's all the adaptive immune system. That's all virus specific. And then the innate immune system is uh, the generalists, right? And really sort of the fire alarm,
Right.
just to
Right.
take some danger. And definitely in COVID, interferon is very important. I'm quite intrigued to see, using these techniques. I'm curious to see if some of these other aspects of the immune system can compensate somewhat for the fact that this virus, to me, if this virus has one superpower, it's its incredible ability to evade triggering interferon for as long as it does, and that has this massive cascading effect to almost everything about the pandemic, essentially. And so I'm intrigued by whether people who have immunity, are there ways that these other cells of the immune system or even antibodies can do things when a viral infection occurs that helps trigger the overall immune system to recognize that something's there even in the absence of type 1 interference. That's where I think for now it fits in.
Well, I think, you're so aptly described, not surprisingly, the superpower of SARS-CoV-2, which I think a lot of people haven't realized that it's so good at shutting down that defense system. Now, you, on the basis of having really gotten this understanding of the mucosal immunity, upper airway, um, does this make you think that the nasal vaccine that we aspire to have is more of a reality? You kind of know what the ideal profile might look like to keep people healthy and resist infections. Do you think this is achievable in any durable sense at high level success with a nasal spray vaccine?
I'm optimistic for several reasons. One is we really saw a lot of different immune memory cell types, that were present. That was encouraging, and seeing the B cell memory durability for at least 6 months,
Mm,
pretty flat line for that 6 months was encouraging. It looks like the
right.
system knows how to keep these cells around, if it wants to for a significant period of time. And we'll have to do more, in follow up. But again, it was encouraging. Third, we had some people who were vaccinated only and some people who had breakthrough infections. And really in the vaccinated only, we didn't see T cell memory in the upper airways. And I actually consider that encouraging because it suggests local exposure does give you the memory and exposure in your arm really doesn't. So I think there is something to improve upon. It can be improved upon.
Yeah.
And lastly, um, I get asked all the time. I'm sure you get asked all the time. Why aren't there more intranasal vaccines or inhaled vaccines, more mucosal vaccines in some way? And I think there's more than one reason. Um, but I tend to be very practical. And I think one practical reason is uh, there's very little to measure to guide you in your vaccine development. If you have 6 ideas or 6 constructs that you think might work in humans as a nasal vaccine, you basically just have to pick one, try something and hoping there's not much you can measure until you get to pretty good clinical trials for what might be the type of immune response you might like. Even so, for example, the flu mist vaccine, right? It's the only licensed inhaled vaccine, intranasal vaccine. And in adults, it doesn't have a clear correlate of protection,
Right.
right? If you get vaccinated with that, your circulating antibody responses don't increase, but also increases in nasal antibody didn't correlate with protection well. So what does that mean? That probably means there's other things going on up there that could be indicative of protection, but weren't being measured before. And so I'm hopeful with these types of approaches now, if you're an intranasal vaccine developer, you maybe have 4, 5, 6, 7, 8 ideas or constructs. If you can try those in a few people and make these different measurements, and you've got your favorite immune profile, right? That you might now you have something to. It's more of an engineering problem. It's not a throwing a dart problem. You're like, yeah, this is giving me the type of response that I like, and I'm going to try and push this into clinical trials. So those are the things that I'm optimistic about moving forward.
Well, I love it because we really need it. And if anybody is optimistic, that means a lot. It's yours. Um, what you've done here has been, quite extraordinary because you defined for the first time, really, the underpinnings of the mucosal immune response, the upper airway. You did it by age, you did it by variant, you did it by vaccine and infection. And most importantly, perhaps for longer term, is you established, what are the desirable features to have, which didn't exist before. It seemed like whatever I read for nasal vaccines, they were measuring some, IgA or IgG, and they didn't get down to the, the memory B cells and the tissue resident T cells, memory cells, and all these other things that you found. You did all this single cell sequencing and flow cytometry. The work is just really fantastic. So, Shane, just in closing, I just want to congratulate you. You made seminal findings along the pandemic. You were the one that really illuminated hybrid immunity, the advantage of if there you don't want to have an infection of COVID, but if you did have that and a vaccine, you kind of had some extra synergy, if you will. But here you've done something, you and your team, unique, and congratulations on that. No surprise that it's in Nature this week, and I'm sure a lot of people will share your optimism that we will have something beyond just shots in the future because COVID isn't going away. There's other respiratory pathogens. And finally, somebody did the right study who knows immunology inside and out. So, Shane, thanks very much.
Thanks, Eric. Very much appreciated, particularly coming from you.